I just want to welcome everyone. We're thrilled to be having our third annual Diversity in Science Lecture Series Symposium. Today, we have an awesome lineup of speakers, including graduate students, instructors, professors, and even our very own Nobel Laureate. Since the inception of DASL in 2020, we have seen tremendous progress and momentum in developing a diverse, inclusive environment across multiple institutions across the country with the ultimate goal of making science exciting, equitable, and attainable. DASL was founded at the University of California, San Diego, where the format of telling one's life and scientific story was first developed. DASL has since grown into a multi-institutional partnership to broaden the reach of our initiative to as many people as possible. Each institution has generated and carried out local, new and creative means to convey the stories behind scientists to pave the way for future scientists from all different walks of life. In other words, DASL portrays the scientists behind the pipette to inspire and guide young budding scientists. The path to a career in science is undoubtedly daunting and can subsequently be inhospitable. However, we aim to create a diverse and equitable and inclusive hub for scientists to collaborate, find community, and thrive together. DASL as an initiative has made progress in this regard, but there is always more that can be done. Without the enthusiastic volunteers who speak, organize, and foster this community, DASL would not exist. For that, we thank everyone from all the institutions for making the DASL community what it has become and for putting the symposium together. And I will just say that we have had over 500 people register across the country, which we're super excited about, and particularly excited to have you all here in person to listen. So without further ado, I will hand things off to William to introduce Dr. Mario Capecchi.
Hi, folks. I'd like to welcome Dr. Mario Capecchi, our resident distinguished professor of human genetics and Nobel Laureate, to the third annual DASL Symposium. When it comes to accolades, he has earned more honors than the number of years I've been alive, which is 31, including the Nobel Prize in Physiology or Medicine, the Kyoto Prize, the Albert Lasker Award for Basic Medical Research, the Franklin Medal, and 32 other honors. As life scientists, you have likely crossed paths with a tool he created, the knockout mouse, which single-handedly changed our perception of genetics, biology, and even medicine. Since coming to the University of Utah in 1973, he has become a cornerstone of the University of Utah, as he's played an integral role in developing the Department of Human Genetics, Biology, and even the Huntsman Cancer Institute. He has accomplished all of this despite the most harrowing challenges that I've heard of, making him an inspiration and role model for many, including myself. And without further ado, I present to you Dr. Mario Capecchi.
Thank you. And first of all, I want to begin by thanking the organizers of the DASL lectureship, and particularly William, for being my host with this opportunity. It's a pleasure and an honor. So, this, where do I point to? So we work on anxiety. The National Institute of Mental Health has reported that the lifetime occurrence of chronic anxiety is greater than 20% among adults, with a higher prevalence in women, 23.4%, and then in men, 14.8%. These statistics are even more disturbing among adolescents and young adults. 38% females and 24% males report the same anxiety. So we generated a mouse that when you disrupt a single gene, OXP8, results both in chronic anxiety and excessive pathological grooming, leading to lesions at the sites of overgrooming. So it is pathological, and the two occur with 100% morbidity. Extensive pathological grooming is very similar to human OCD, spectrum disorder, trichotillomania, which is manifested by pulling of hair. This shows that it is pathological, that is, they continue to groom until they have lacerations at the sites of overgrooming. As in humans, both behavior pathologies, chronic anxiety and pathological overgrooming, show strong sex bias, with female mice exhibiting more aggressive phenotype. This is shown in this slide. This is simply a heat map where you overlap about 20 different grooming patterns in the respective mice, and then simply give you a heat map. And what you can see is the females have much more aggressive phenotype than males. Is excessive grooming anxiety behavior in HOXP8 mutant mice related to production of female hormones? And the answer is yes. We've done this in two different ways. One is to remove ovaries, and the second is to treat with trilostane, which inhibits the synthesis of female hormones. And what you can see is that the mouse, then the grooming behavior in a female mouse goes down to male levels. You can do the converse. We can inject progesterone and beta-striol, the precursors to estrogen, and show increase in pathological level to the female level. And that's shown here. So there's a male mouse that we introduce to hormones that goes up to female levels, and it's controlled just a ordinary mouse, not a mutant mouse. So multiple assays are used to assess anxiety, elevated maze, open field tests, and light and dark boxes. This shows the elevated maze. It's two platforms that are orthogonal to each other. One of them has no walls next to it, the other is enclosed in walls. A normal mouse will explore both platforms equally. An anxious mouse will obviate the open arm. And what you can see is females are very anxious relative to males. Here's an open field test where you have four walls. Then the mouse is put in. A normal mouse will explore the entire parameter of that field, whereas an anxious mouse will obviate the center where they feel more comfortable. Again, you can see HOXP8 mutant mice obviate the center. And all of these can be quantitated. We can quantitate in terms of how much time they, for example, spend in the center, or in a light dark box. There's just two boxes. One has a light in it, one has is dark. And a normal mouse will explore both equally, and an anxious mouse will stay away from the lit one. So these are all assays essentially where they're obviating uncomfortable situations. In all of these tests, HOXP8 mutant mice stand high with respect to chronic anxiety. Now, what is the causal agent? The cell type that's responsible for the behavioral phenotype. And we approach this by doing cell lineage analysis. And that's shown using Cre-Log-Speed. You have a Cre driver in the HOXP8 locus. So wherever Cre is expressed, then it activates GFP, which is ubiquitously expressed locus, but is stopped by a transcriptional stop signal, which is flanked by LOXP site. Wherever Cre is expressed, that will be taken away and cells turn green. And here we obtained an enormous surprise. The only GFP labeled cells in the brain were microglia. We're all familiar with microglia. This shows you the actual experiment. So here we have essentially YFP, and then you have CD11B, this is the pan-microglia marker in red, and then you see coincidence in the third one, and also see that only about a third of the microglia are actually labeled with the cell lineage marker. It's not all microglia, just a third of the population. Microglia are immune cells. Okay? They essentially remove debris and then also fight pathogens. That's a classic view of what microglia did. But what we're suggesting is something quite different. And that is that HOXP8 cell lineage microglia regulate specific neural circuits that are dysregulated in the absence of HOXA8 function. And that results in pathological behavior. So there's a direct link between microglia and specific neural circuits, which then give rise to behavior. Now, what is the ontogeny of microglia? It has been posited that all microglial progenitors are generated during the first wave of yolk sac hematopoiesis and then directly migrate into the developing brain at E9.5. This is beautiful work of Gene Ho and colleagues. Okay, so we wanted to see, look both at normal microglia, non-HOXA8 microglia, as well as HOXA8 microglia. So we made two colored mice in which all microglia are labeled with a fractalkine receptor, okay, that's expressed in all microglia, green, okay, by insertion into that gene. And then HOXA8 microglia are now labeled red, digit male, and again, by having a cassette that makes it dependent on cream. And then we use HOXA8 iris cream to drive that activation. So the first thing we did was simply to look, when do we first see HOXA8 microglia in the brain? Okay, green, all microglia, yellow, only HOXA8 microglia. And remember, they started getting into the brain at E9.5 station. By E10.5, still no yellow cells. And then at E12, we start seeing yellow cells in the brain. And then they continue to go up until about P8, 8 days after birth, and then plateau off at about 30%. So this could be explained by two possibilities. One is that a subpopulation of microglia in the brain now turn on HOXA8 and become HOXA8 microglia and become yellow. The other is that it's an independent, separate wave of hematopoiesis that gets into the brain starting at E12.5 gestation. We can distinguish between these two possibilities simply by looking when HOXA8 is actually expressed. What I've showed you so far is cell lineage markers. You turn it on, it stays on forever, okay, even when the gene is turned on. But when the transcript is made, and here we're looking at transcript in different parts of the hemoplastic lineage. In the yolk sac, at E7.5, there's no HOXA8 expression. Then we see high expression and then tailoring off fairly quickly. AGM, E10, we start seeing expression, and then that starts tailing off. In the fetal liver, there is expression there, but it's very, very low. But what's important is in the brain at E9.5 all the way through adulthood, there's never a single transcript made. Okay? That rules out the second hypothesis, or the first hypothesis. Okay, can't be a subpopulation since HOXA8 is never expressed. So that leaves open then the second population of moving. Okay, so that rules out the first hypothesis, and that leaves the second. So now we have essentially two sources of microglia, non-HOXA8 microglia. Start at E7.5 in the yolk sac, okay, and then go into the brain at E9.5 when the vascular is formed. Okay, HOXA8 microglia are also born in the yolk sac, but a day later, E8.5, and then migrate now to the AGM, get amplified 16-fold, then to the fetal liver, get amplified 280-fold, and then start migrating into the brain. Okay? Very different ontogeny. Okay, so what are the similarities and dissimilarities between these two populations? So TMEM19, SAL1, and P2Y12 are what are classically known as markers for microglia. And all three of these markers are equally expressed at high levels, both in non-HOXA8 microglia and HOXA8 microglia. So that says that HOXA8 microglia are bona fide microglia by that criteria. Okay. Do HOXA8 behave like microglia? And hopefully the film should work. So what you have here is a field. We're looking, this is live imaging in a live mouse using 2-photon microscopy. what we're going to use is a laser to kill a neuron right in the middle of the screen. Okay, and what you're going to see there is reaction. You see Hoxsmeade microglia yellow over here, non-Hoxsmeade microglia over there. What you're going to see is a convergence essentially of all the filopodia going to the site of injury. There's a, and now see it goes right there, and they arrive there at exactly the same time. Okay, so by this assay, they're indistinguishable. Another thing that microglia do is it's an active protein. As you all know, you make twice as many connections as normal, and then you weed out the weaker connections, thereby reinforcing the strong ones. And that's an active protein. This is an assay worked out with Stevens. This what she did was to progressively label the beta subunit of prototoxin, which has extremely high affinity for synapses. Okay, so then you inject that into the eye, it travels retrograde into the brain, into the thalamus, okay, and then that gets associated with synapses. Then the microglia start clearing out the bad weak connections. Okay, what you see then is synaptic pieces inside the microglia. And as you can see here, for example, over here, there's a green cell, it's non-Hoxsmeade microglia, a yellow cell, which is Hoxsmeade microglia. Then you can see the puntate, which then are the colotoxin bound to synapses. What you can then you simply do is count hundreds and hundreds of these cells, and what you can see is that Hoxsmeade microglia are as good, if not better, than non-Hoxsmeade microglia. So again, in this assay, they do equal or better. Okay. Molecularly, the two populations are also very similar. There are only 40 genes that distinguish the two populations that are more highly expressed in one or the other of these cell types. Now, are defective Hoxsmeade microglia causative? We've shown that they're there, but the question is, are they really causative for chronic anxiety and pathological overworkings? And Don, in our lab, approached this not by simply doing transfers. Okay, what you can do is get rid of all the microglia in the brain, and we'll show you how to do that. And then once all the microglia in the brain, then you can inject new ones and then allow them to take over. Okay. And this is how you get rid of the microglia, the endogenous microglia. You use fractal receptor I-secrete, then CSF1 are required by all microglia for survival. You flash that and then inactivate it in all microglia in the brain, and then that gives you a clean slate. And then you do the injections and wait for a while, 4 to 6 months. And so transfer the progenitor cells, both sides of the brain. And then here you can see very good engraftment if we use the bone marrow source as a wild type or a mutant. Okay? And now we can compare and see what happens to these mice in terms of phenotype. And that's shown here. So you have here a wild type mouse, no grooming. You have, if you put in mutants microglia, Hoxsmeade microglia, then you have extensive grooming, removal of body hair, twice as much time spent grooming if it's mutant, and also higher levels of anxiety because they're spending most of their time in the throat guard. Okay, so this proves that putting a purified cell population into the brain and get the phenotype, they must be the causative agent. Okay, so that proves indeed that they are the causative agent. Okay, so now we're positioned to start actually looking at how this whole system works at the molecular level. Okay, and we've done that by meaning Naveen, who's in the audience. The first step is to see if we can modulate anxiety and grooming by optical meds. Okay. And the first question you ask yourself is, how is that possible? Microglia are not neurons; they cannot make action potentials. Okay. So what do we mean even by optogenetics of microglia? What we mean is simply they function. The channel works as a putting in cataracts. Okay. That depolarizes the membrane, that changes the cell, and then they can respond to different things. Okay? So we're simply looking at depolarization of the cell membrane. And that's what we said here. So requirements are HOXP8-CRE. They drive this HOXP microglia. CRE-dependent channelrhodopsin, that's an AI32 allele, which you can actually get from Harvard. Laser activation in very specific regions of the brain, and then see what happens. Okay. But before I show you the experiments, I want to just show you a few controls. Okay, that shows that this reaction is highly specific. That is, what we're going to be looking at is simply spatial specificity. That is, some places work, some places don't work when you activate the laser. Okay. Cellular specificity is specific to HOXP. And it's also been reported, and that's always been nagging at us, that AI32 has some leakage. Okay, in non-CRE expressing cells in the neurons, there's very low level of leakage. The question is whether that could also be responsible for what we're seeing. So those are the questions we're going to address shortly just now. Okay, well, that's good enough. It doesn't want to advance.
Is that the next one?
Yeah.
Perfect.
Good. Okay. So what I will show you is these regions of the brain, dorsal medial striatum, medial prefrontal cortex, and so on. Those are places that work, but there are other regions that do not work. And I'll just give you a couple of examples. Here is the primary visual cortex. When you put the laser in the primary visual cortex, exposes the microglia in that region, and then what we can see is no reaction. There's no grooming behavior, no grooming dots, and so on. Everything is zero. Here is we're looking in the hindbrain at the nuclei that are responsible for controlling essentially breathing. And again, if you do the experiment, here we go and see zero activity. Okay, so there are many regions of the brain that you can expose laser to, and nothing happens, even though there are microglia there that could be activated. Okay. We can also epigenetically activate other cells, astrocytes, using GFAP iris screen. Okay, and then again, AI32. Okay, and when we do that, again, zero. And in the striatum, in this case, was in the striatum, and again, no moving behavior. Okay. So those are controls of the spatial specificity. Finally, it has been reported that the AI32 LDL leaks. Okay, so you have to be cognizant of that and see what happens. The leakage is very low. That is, the people that reported it could only see it in mice that are homozygous to AI32. Okay, if you look at a heterozygote, just a twofold reduction, then it's below detection level. So it's really low level. And nonetheless, it's there. And so what we've done is simply, first of all, look to see whether we could see it. Okay, so here we're looking at neurons in purple, and then microglia, all microglia are red, and then YFP. I should say the AI32 allele has the YFP gene in it. Okay, so if you activate it, then cells turn green. Okay, and then we can see green cells. And what we're looking for is neurons that have green that are not associated with microglia. I'm showing you here a couple of sections, but we've looked at hundreds of sections. Okay, there is no leakage observable. But even in mice that are homozygous, but if we leave out HOXP8 iris screen, then do not show any activity. Okay, and not having HOXP8 iris screen in microglia should not affect leakage in neurons. Okay, and then you stretch of the imagination. So even if it's homozygous where it can be detected, still no behavior. Therefore, that is not responsible for the behavior. Okay. So we don't have to worry about that. So now let's move on to the actual experiments. So here we're looking at the dorsal median striatum. We can use C57 black 6 mouse as a control. If all of the mice that we've used are highly backcrossed to C50 black 6. So it's a control among others that we use. So if you have C57 black 6, you turn on the laser in the DMS, then nothing happens. If you turn it on in the dorsal median striatum, then you get grouping activity, robust, okay? Big, huge difference. The protocol is to take movies of the mouse 2 minutes before we apply the laser, then turn on the laser for 2 minutes, then turn off the laser and look for 2 more minutes of recovery. Okay, so that's what these experiments are. So you see essentially there's pre, during, and then after. Pre, during, and then after. Okay, and what you can see is the decay is immediate, okay, but it's pretty fast and fairly complete. Okay, and then you get robust when the laser is being activated. And here's the same experiment with the medial prefrontal cortex, and again, a very strong, robust grouping. Nothing in C57 black 6 because they don't have AI32 or NHOXB8 or screen. Then in the mouse that has both markers, then you get grouping and so on and so forth. And again, what you can see is a very good response, pre, during, and then after. Very good decay. And in fact, the medial prefrontal cortex is more sensitive than the dorsal median striatum. And this is females. When we've looked at males and females, we can't distinguish the two. So in terms of this assay, which we're not creating chronic levels, just higher levels, there is no sex bias. Okay? The sex bias is for the pathology. The next region we looked at is the amygdala, and particularly the ELA and the CA, the basolateral amygdala and central amygdala. And here we're looking at when we did these experiments, there was no grouping, but elevated anxiety. And in this case, we're measuring the elevated maze. What you can see is they're exploring both. Turn on the laser, obviously open. Turn off the laser, they start exploring both. Okay? And you can quantitate this and show here essentially, you can turn the laser off, turn it on, and boom, let the drops going into the open arm. And finally, the other thing I want to point out is that the distance travel also goes down. And the reason is, and this is known from other work, is that one of the things that you induce is freezing. That's essentially an extreme effect of essentially being over anxious. So you just freeze, and therefore you don't go as far. So that's another characteristic. And here we're using the open field test. Again, in the BLA and CEA, we're operating the center pre to explore the entire activity. And again, we look at freezing now and see essentially that goes up, and particularly with respect to CEA, the central. Okay, your controls essentially you're using like 6 mice, whether the laser is on or off, as care to expose the entire surface, and then similarly it has no effect essentially on time spent in the center with respect to turning off and on the laser. The final area we looked at is the VCA1 region of the hippocampus. Okay, and here we got both grouping and anxiety. Okay, so in the BLA and the DMS and MPF medial prefrontal cortex only grouping, amygdala only anxiety, and then the hippocampus both. Okay, and here we're showing the anxiety levels. Look at it before the lasers turn on, they explore both, and then they restrict themselves in the closed arm when the lasers turn on, and so on. And here we can follow essentially what they're doing with respect to the closed arm, it goes up, and in terms of the open arm, it goes down. This one looks at open field, within the control up above, and down here is the obviating the center with respect to in the VCA region. Okay. And then finally, here's the grooming, again, robust grooming. I should say that the hippocampus is the most sensitive. And then we can see that particularly in the laser power utilizing. In the amygdala, we have to go up to about 10 millivolts, whereas here we're looking at 2 millivolts. So they're really sensitive, which means either that they're more sensitive or they're more microglia in that region. Okay. So thus far I've showed you that such as the results of optogenetic activation of OxP8 microglia specific regions of the brain. What happens if we optogenetically activate non-OxP8 microglia? Unfortunately, we cannot do that experiment. And the reason is we don't have a genetic marker that distinguishes non-OxP8 microglia from all other microglia. But what we can do is activate both. And then compare it to just the activation of OxP8. Okay, and subtract that out and see what's happening to non-OxP8 microglia. Okay, so we use ABL1 create to activate both microglia subpopulations, which is expressed in both microglia subpopulations. Okay, and what the expectation was, either we would see no differences. Okay, so that means that non-OxP8 microglia are not participating in either group or anxiety. Or alternatively, we see higher levels of both, which would tell us that then both are microglia participating. Okay, those were the expectations, and the experiments were actually mind-boggling. Neither is right. Okay, you obtained a completely unexpected result, and that is eradication of grouping and anxiety. Okay, so that was a real surprise. And what it means, we have to completely revamp how we're thinking about this system. What it means is the two populations. Okay, I'll show you the experiments. Just that they are zero. Okay, here's the DMS, which normally will give you lots of grooming, nothing. Activate both populations. And here is the medial prefrontal cortex. Again, you have to activate both populations. Zero, nothing. Okay. Remember, I showed you the first figure of these experiments. You got a huge amount of movement when you turn on the laser. So these observations simply say we've got to think over what's going on. And what's going on is that the two populations are working in opposition to each other. It's not that one's doing something and the other's doing something, but they actually do the opposite thing. Okay. And what we're seeing is, and the reason we're seeing it this way, what we're saying is that OxP8 microglia are brakes. And their role is to down-regulate grouping and anxiety. Whereas non-OxP8 is the opposite. They're an accelerator. Their role is to upregulate it. And then together, they can reach an equilibrium everywhere they want. Okay, so they can set the point with respect to the body or homeostasis of the mass. So this model also provides a reason why do you have two populations to begin with. I mean, that one of the things you might have asked yourself is why, if one's good, why not bother? Why have another one? Because now you have much finer control with respect to setting where you want the level of anxiety. Remember that low levels of anxiety are good. Okay, they get you going, they get you excited, they're what drives you. It's simply the chronic levels of anxiety. So you simply want to set the situation for what's happening to you at that particular time. Okay, so this made a very strong population of prediction. If we had a mouse that only had green microglia, should have higher levels of anxiety because there are no brakes. Okay. And indeed, this happens. So here we have essentially, remember again, because we can wipe out, we can put anything back in we want. So we can put in normal microglia or OxP8 microglia or green microglia and see what happens. And what's important is here. Okay. These are controls, these are heterozygous. Okay. So I don't want control because there's still activity there, we don't destroy it. But these are the important ones. And whether the source is normal non-Hodgkin microglia or mutant microglia, remember, a Hodgkin microglia should not affect green microglia. Okay, Hodgkin is never expressed in non-Hodgkin microglia. Okay, so these two populations show very high levels of decline over in this case group. Yeah, whereas here, if you just put in wild PA, should be that before. Okay. So this essentially, and nobody would have predicted this. And therefore, we feel confident that this model where those two populations work in opposition to each other are actually robust. So in summary, what I've showed you is the two microglia populations, that activation, inactivation microglia causes specifically reduce elevated anxiety improvement. And finally, the model where essentially we have these populations doing opposite things. One function, Hodgkin microglia function as a brake, whereas non-Hodgkin microglia function as an accelerator. So the organizers of DASL, I'm still doing that, DASL insisted that I talk about personal stuff. And that's a bit the scientists say. They love talking about science. That's why they're scientists. But here it is. Now, I think all of you are aware that science is a very human experience. And therefore, it's not surprising that what we do, what captivates us, is affected by our nurture. And I think I may be considered to have a chaotic beginning. I was born in essentially at an early life. I started at 4. I lived in the streets of Italy. I was responsible for everything. And what you're mainly concerned with is food. Everything else is secondary. What you're really interested in is where you're going to get the next meal, and nobody's going to give it to you. You're going to have to produce it yourself. So there's been a film produced, it's called Vision. It's made in Italy, and it's now out. And those of you who are good at streaming can probably find a way of getting into that film. Okay, this is my mother, age 19. She was a poet, and she got her training at the Sorbonne in Paris and became a lecturer there. And there she became politicized, started making pamphleteering. This is in the late 30s against fascism and Nazism. And she knew that was a dangerous activity. So then she moved essentially up to Tyrol and spent several years, and that's where I was conceived, in the Tyrol. And she knew, but she continued her pamphleteering from up there. And she knew sooner or later the Nazis would get her to the South. And what she did was to simply give all the money that she had to a farming family that she knew that if she was picked up, I was to go to the family. And she was picked up in the spring of 1941. And then I was sent to this farming family. And where my mother ended up was in Dachau, which is a concentration camp just outside of country. And I should say that what she experienced is, beyond my own imagination. She would never talk about it. And, she's an attractive woman, and she was in hell, essentially. Fascists were just pigs. This is a picture from Dachau, and that's the way they treated people in Dachau. So in 1945, my mother was still alive, which is actually quite surprising. But she was liberated by the American troops that arrived there. And then she spent essentially 2 years looking for me. When I ended up, I was in the hospital with typhoid and malnutrition. A week after she arrived, we were on a boat sailing for New York. My uncle had sent her money for the boat trip, and we got on a boat and then traveled towards US. This is a picture of me about a week after arriving. I was 9 years old, had never been to school, had never learned how to read or write. And then Monday, so I arrived on Saturday, went through Ellis Island on Sunday, and Monday I was going to school. So that's what this says. So, ironically, my uncle, who was a physicist, brought a commune. So I'm going from a completely asocial situation to a commune where I had 75 parents. They didn't bear with me much. It was amazing. This is my uncle. He's a physicist. He's an outstanding physicist. Electron optics was his field. He made actually the first electron microscope. Here he's working on a project he was ashamed of, TV. No TV in our house. So back to science. The question is, what characteristics are attributable to my science? And a prominent feature is that we periodically exchange topics every few years, every 10 years or so. Started out in molecular biology. At the time, molecular biology was just being born. It was heady. It was amazing. And the reason it worked is because of the influx of scientists from all different fields: physics, chemistry, biology, medicine, and so on, all working together to actually solve problems at the molecular level, as opposed to phenomenological. Nuremberg had just published a paper saying that if you stick poly-U into protein synthesizing system, cell-free system made from bacteria, you make polyphenol. And I looked at that and I read that just the summer before going to Harvard Graduate School. I was impressed. Then what we did was to simply not use poly-U, but simply take viral RNA, put it in the protein synthesizing system to see whether we could make viral products. Particularly, this is a simple virus, which had only two proteins, the code protein and the polymerase. Okay? And it made both, and they were functional. And that was amazing. Okay? You stick those RNAs in a cell-free system, and out come real proteins. And once you had that, then you could study anything you wanted about protein synthesis. Nothing was known at the time. They knew the protein synthesis occurred in ribosomes. For example, they thought, for example, that every ribosome was making a different protein, and so on. I mean, the level of what knowledge was, basic. But any question we answer was a breakthrough. And so I used the system to study initiation, how the protein starts, how they elongate, and how they terminate, as well as a mechanism for genetic suppression. Okay, the next stage was gene targeting. And this was quite an issue. That started here. I was thinking about it previously, but I decided Harvard wasn't the place to do it. Maybe Utah was. So we came here and we said, we're going to make essentially specific mutations in a mouse. And people just thought we were nuts, particularly the NIH. And they said, impossible. Literally, they said, impossible, no money. Okay? So what do? You become resourceful, you use another grant and dump it into this project. Big risk. If you didn't succeed, I would be scrubbing floors. But I knew that it was big, because mutations are at the heart of genetics. Without mutations, genetics is worthless. Okay, so if we could create mutations, and the problem with the mouse was that everything was occurring inside the womb. You can't see what's going on. Nonetheless, but there were mutations, code color mutations, so people saw those in mice, but that was the limit of the experiments. And so what we wanted to do is to knock out a gene, for example, and see whether we perturb brain development of cancer. and improved NIH law. Okay, then next thing, question is, now you have this technology, what are you going to do with it? Okay, the smart thing would have been immunology. Because many genes have been discovered, but they have no idea what they're doing. But I wasn't an immunologist, I wasn't interested in immunology, but I was interested in developmental biology. How do you create something starting with 2 cells and make something as complicated as us? Okay. So I decided to go into developmental biology and specifically Hox genes. Ed Lewis had knocked out Hox genes of testosterone, and amazing things happened. You had legs coming out of your head, huge transformations. And then just at the time that we did got gene targeting, the first inkling that Hox genes existed in other organisms, specifically mouse, just opened up. So the question is, what are they doing in a mouse? So that's what we pursued. Well, the next stage was childhood cancers, sarcomas. Why childhood cancers? Simply because not many people were working on it. They're not as common as carcinomas, and therefore all the money was going into carcinomas and not into childhood cancers. But they're extremely aggressive. For many of those diseases, the prognosis was that 80% of those children would die 5 years after first presentation, period. And then finally, we came to which is a stage we're at right now, is neuroscience. And one question I ask you, why did you do this? Why do you keep jumping around? And one is simply, I love thinking about new things, okay, and being invigorated. And one of the things I did was simply, the first time I say, okay, I'm going to go into this field, I teach a course in it. Because that makes me really learn the field and read the literature, because or else you guys would trip me up. So I think it's simply the curiosity of looking at a new field, and you always start at the bottom and you have to work your way up. There's no question about it. And that's what you do. But one of the things is, once you're naive in the field, you do not know what questions you should be asking. People in the field know which questions you should be asking, and somebody's naive. So we may ask questions that other people aren't addressing. So I'm going to spend a couple of minutes with the structure of DNA, because that's everything. Everything I do, everything many of you do, is just dependent on DNA. And I think all of you are familiar with DNA, and every time I look at it, I'm amazed. It's a machine that's just made for archiving information, huge amounts of information. And it's a very simple machine. It's conceivably simple, but very profound. And the secret of DNA is simply here. And actually, Watson worked this out. He just made paper cutouts, cardboard cutouts of the base pairs, and then just started shifting them around and around and around. And he found that there was only one configuration where he maintained essentially the ribosphosphate backbone at equidistance. And that is when T, if there's a T on one strand, then the other strand had to have a name. If there's a C on one strand, then the other had to be a G. Okay? And that's the whole secret of DNA. Because if you know the sequence of one, you obviously can read out the sequence of the other. And that immediately told you how that whole system worked. And that's simply you separate the strands and each form a template for a new strand. And it's amazing. The specificity is enormous. Makes one mistake in 10 over 10 to the 9th base pairs every time it's replicated. So here you go from mother cell to the daughter cells, and each have the same information about it. This is Jim Watson at age 25, Crick at age about 40, working out the structure of DNA, 1953. Watson ended up being on that earth. This is also a person, Rosalind Franklin, who should have been in the Nobel Prize, but she died before that, and she contributed essentially, not willingly, the structure of the weave from the DNA that this model is dependent on. Here's Watson at the later stage, looking a little bit more. Another question I want to pose to you is simply, do mentors matter? And I was very fortunate to have Jim as a mentor. But I think so, and I'm going to show you a scientific tree lineage. At the top, there's a guy named Giuseppe Levi, and he had 3 students, Gloria, Del Becco, and Liga Montalcini. All 3 won the Nobel Prizes. And then you look at Del Becco, he had a student named David Baltimore. He won the Nobel Prize. Then there's Watson, and he had a guy named Capecchi, and he had won the Nobel Prize. This doesn't just happen by chance. There are other trees like this. And that shows you that mentorship is really important. So I started by saying that science is an extremely individual process, and that each scientist is different. But oops, I'm ahead of myself. Okay, I'm going to just show you a couple pictures of the ceremony, since we're talking about it. So here's the stage. It's set up very purposely. Here are the Nobel Prize winners for that year. Here is the people that are involved in making that decision. And here is the royal family. Here's a closer shot. You can see what people are looking at. The laureates. And here is the royal family. And what you do is very simple. You simply keep an eye on the king. He stands up, you stand up, he sits down, you sit down. This follow him, and then everything will be okay. Here's receiving the prize. Then here's an intimate dinner of 700 people after the ceremonies. Okay, that's the future queen. And this is the Nobel Prize in Physics from France of that same year. Okay, so here it is. I emphasize individualism in the scientist. But what I want to also emphasize is that science is a communal process. We do it all together. And the science has no boundaries. You can do good science anywhere. And then what's important is that we do work together. We add knowledge that previous people have generated. We add, and then people will add to what we and then I want to also emphasize that the group that we have right now is smaller, but it works beautifully together. Here's our current group. I want to emphasize certain people, not at the exclusion of the others, but the genetics is right here. Don has done the transportation experiments, and there's all our genomics, which we rely on for the rest of the projects, and then all the rest should be. But with that, I'd like to